Okay, Assalamualaikum and selamat sejahtera. So now let's proceed, okay, to the subtopic 4.1, okay, which is about Mendelian genetics, monohybrid and dihybrid. So in this video, we focus on monohybrid. Okay, so first of all, what is monohybrid? It is refers to the breeding that involve a single characteristic determined by one gene. Okay, so the keywords here, it will involve single characteristic. So example, high, seed color and flower color. So kalau kita tengok dekat sini, maybe the genes, okay, is control the height. Okay, genes control the seed color and also genes that control the flower color. Tapi kalau dari segi konsepnya, monohybrid ni dia cuma akan melibatkan ataupun dia akan involve single characteristic that determined by one gene. Okay, so for monohybrid, actually you have to relate with the Mendel's first law that we call as law of segregation. Okay, so what is law of segregation? It is two allele for heritable character separate or segregate during gamete formation and end up in different gamete. Okay, so secara ringkasnya, law of segregation. Actually, segregation refers to the separation. So, apa yang berlaku kepada two allele, okay, for a heritable character, okay, dia akan separate ataupun segregate bila sahaja berlakunya gamete formation and then it will end up in different gamete. Okay, so nanti kita akan tengok contoh. Okay, so kita tengok dekat sini dulu. So based on Mendel's first law or law of segregation, the first one, each trait or characteristic of an organism is determined by a pair of allele. Maksudnya sesuatu trait, okay, ataupun karakteristik untuk sesuatu organism tu dia akan ditentukan oleh a pair of allele. Allele tu mesti kena ada dalam sepasang. Okay. And then each pair of allele segregate or separate during meiosis. Okay. Yang ini kita dah discuss dalam chapter 3, which is kalau untuk meiosis berlakunya separation of the homologous chromosome dan juga separation of the sister chromatid. Okay. Dia akan berlaku pada anaphase 1. Okay, dan juga anaphase 2. Okay, and then third, only one of each pair of alleles can be present in a gamete. Maksudnya, untuk setiap gamete, kalau dia adalah law of segregation, dia cuma akan ada satu allele sahaja disebabkan oleh pair of allele tadi telah segregate ataupun separate. Okay, so look at this diagram. Okay, so for the example here, let's say, okay, for this cell, okay, dia ada chromosome like this, okay, tetapi dia punya genotype dekat sini adalah A besar, A kecil. The first chromosome dia ada dominant allele A besar, second chromosome dia ada recessive allele A kecil. So, apa yang akan berlaku? Bila berlaku segregation of these two alleles, kalau awak tengok dekat sini, yang ni kita panggil sebagai homologous chromosome. So, this homologous chromosome will be separate or segregate during anaphase 1. So, what happen? Both of these chromosome will be separated lah. So, dalam satu sel ni, dia akan ada chromosome with allele capital A besar. Dekat sini, dia akan ada sel which is kromosomnya adalah contain allele A kecil. So, bila diteruskan dengan proses meiosis 2, which is during anaphase 2, so segregation of these two allele occur. Maksudnya segregation of these two allele akan berlaku melalui separation of the sister chromatids. So sister chromatid ni akan terpisah lah. So that's why dekat sini awak tengok sister chromatid ni akan separated. So untuk setiap satu sel, okay, ada dua kromosom like this. So akan ada allele A besar dan juga allele A besar. Manakala for the second one, since dia punya sister chromatid is already separated dengan allele ni adalah A kecil. So, bila dia separate dekat sini, okay, so setiap kromosom ni akan ada allele A kecil. So, secara konsepnya, kalau awak relatekan dengan law of segregation, maksudnya these two alleles will be separated or segregate during meiosis and it will end up in different gamete. And then setiap satu gamete dia cuma akan ada satu allele sahaja. Maksudnya kalau kita ambil contoh this gamete, dia cuma akan ada allele A besar. Yang ni pun sama, dia cuma akan ada allele A besar. So that's why kalau awak nak tunjukkan genetic diagram, okay, let's say this is the heterozygous tadi, A besar, A kecil. So during gametes formation, so the gametes will be, satunya adalah A besar, satu lagi adalah A kecil. So ni kita panggil sebagai law of segregation. Okay, so now we look at the monohybrid cross. So what you have to take note about monohybrid cross is you have to know the ratio when you do the self cross, okay, of the monohybrid, and also when you do the test cross of monohybrid. Maksudnya awak mesti kena tekankan berkaitan dengan ratio. Sebaik sahaja awak dah lakukan self cross dan juga test cross. Okay, so kalau kita ambil contoh dekat sini, the example here is plant high. Okay, so for plant high, it has two traits. The first one are tall, second are short. So, untuk tall ni kita refer kepada dominant phenotype, while for short we refers to the recessive phenotype. Okay, so for tall, okay, so kita boleh gantikan ataupun kita gunakan simbol huruf T lah. So huruf T dekat sini, kalau kita ambil contoh yang pertama, capital T ataupun T besar, it will represent dominant allele for tall. While for the small t here, T kecil, it will represent recessive allele for short. Okay, so first of all, firstly, you do the true breeding first. So remember what is the meaning of true breeding? True breeding, it is the crossing between parent which is both are homozygous. So means that dia akan melibatkan crossing between homozygous dominant parent and also homozygous recessive parent. So homozygous dominant is T besar, while homozygous recessive is T kecil. So, bila awak dah tulis genotype for the both parent here, so mesti kena tulis lah parental genotype double dot like this. Okay. And then second, you have to show the gametes. So, for the first parent, which is homozygous dominant, since dia bawa allele yang sama, which is T besar, so awak just turunkan satu lah. So, gametes dia akan jadi T besar. Okay, why the other one? Homozygous recessive parent. Okay, so the gametes, in its gametes, dia akan ada T kecil. Tapi kalau kita ikutkan, bila kita follow law of segregation, bila sahaja berlakunya separation of these two allele, dia akan ada T besar dan juga T besar, kan? Tapi since dia bawa allele yang sama, so kita boleh assume lah satu sahaja. So, bila awak buat crossing in between the allele T besar dengan T kecil ni, so kita akan dapat F1 genotype. So, the F1 genotype here, it will be T besar, T kecil. So, this is heterozygous. Okay, and then awak mesti kena ingat lah F1. F mesti kena besar, 1 ni kena kecil. Okay, so, what is the F1 phenotype? All F1 are tall. Ataupun awak boleh mention all offspring produce are tall. Since, okay, the genotype of the F1 all is heterozygous. Okay, and then next, you proceed with the self-cross. Okay, so self-cross means self-fertilized, okay, between the same generation. So, kita dah dapat tadi, F1 generationnya adalah T besar, T kecil. So, bila kita nak buat self-cross, F1 cross F1, so tulis macam ni, double dot. Okay, and then just copy lah, T besar, T kecil, cross with T besar, T kecil. Okay, and then gametes. Okay, so if you look at here, since the gamete here, dia ada T besar dan juga T kecil, kita tengok dia punya allele dia akan ada T besar dan juga T kecil. So, both of these two allele will be separated kalau kita refer kepada Mendel first law. So, in the first gamete, dia akan jadi T besar. Second gamete, dia akan jadi T kecil. Same as the second one lah. So, dia akan ada gametes yang ada T besar. Satu lagi gametes yang ada T kecil. So, bila awak buat crossing untuk dapatkan F2 generation, so the genotypes of F2 will be, okay, so awak just kena crosskan lah T besar with T besar. So, awak akan dapat homozygous dominant T besar, T besar. And then, for the next one, T besar, T kecil. So, awak akan dapat heterozygous. Okay, and then, for the third one, T kecil, T besar. So, tulis T besar dululah. Sebab T besar ni adalah dominant allele. So, dia pun akan menjadi heterozygous. And the last one, T kecil, T kecil. So, this one adalah homozygous recessive. Okay, so that's why for the genotype ratio, F2 genotype ratio, so you have to write lah 1 homozygous dominant, T besar, 2 heterozygous, T besar, T kecil, 1 homozygous recessive. You have to write. Okay, it's genotype. Jangan tulis 1 to 1 sahaja. Okay. And then for F2 phenotypic ratio, so since kita ada 1 homozygous dominant, 2 heterozygous, which all of this, okay, the 3 of this, it will represent the dominant phenotype. So that's why for the phenotypic ratio, it will become 3 tall. So tulis tall. 2, 1 short. Okay, so this is for the test. So this is for the self cross. Okay, so now we look at the test cross. Let's say you want to do the test cross from the F1 generation. Okay, so remember again, what is test cross? Actually, it is the crossing that involve individual that has dominant phenotype with unknown genotype with homozygous recessive. So you have to cross it with homozygous recessive. So since from F1 generation, kita dah dapat dia punya genotype ni adalah heterozygous. Okay, so you just do the test cross of the F1. So tulis sahaja F1 in bracket test cross. T besar, T kecil, you have to cross it with homozygous recessive, T kecil, T kecil. Okay, and then what is the gametes for heterozygous? Okay, so it will be the first gamete, dia akan ada allele T besar. Second gamete, dia akan ada allele T kecil. Okay, and then for the second parent, for the second genotype here, okay. So, since dia adalah homozygous recessive, so the gametes akan ada T kecil. Okay, and then you show the crossing, okay, by which kita nak dapatkan F2 genotype. So, dia akan jadi the first one T besar dengan T kecil. So, it will become heterozygous, and then the second one it will become homozygous recessive. Okay, so what is the genotypic ratio for F2? So dia akan ada 1 T besar T kecil to 1 T kecil T kecil, and then for the F2 phenotypic ratio, so it will produce 1 tall to 1 short. So as a conclusion, kalau awak tengok test cross for monohybrid, if the genotype is heterozygous, so its phenotypic ratio, you will get 1 to 1. Okay, so you have to remember the ratio lah for both self-cross and also test cross.